Okay, I'd like to welcome everyone to this talk. Um, we have quite a few people here, and so I'm going to ask our co-host to go ahead and mute microphones during the talk. If you have any questions, please feel free to put them in the chat box, and we'll get them presented to Rich at an appropriate time, probably at the end of the talk. Rich is going to be talking for about an hour, and then we're going to just let whoever would like hang around for another half hour after that to ask any questions that they have. We'll be opening the mics up at that point. I'd just like to take a little bit, a moment to introduce Rich. He is the director of our facility, GTAC at MGI here at Washington University. Rich brings over 25 years of experience in medical research in both academic and industrial settings, most of it in the development and use of high throughput genomic technologies such as microarrays, high throughput PCR, as well, of course, as next generation sequencing. He and his teams have successfully brought these technologies to bear on the discovery of novel drug targets and their modes of action, biomarkers for patient stratification, development of preclinical models, and ascertaining the causes for drug failures. While at WashU, in addition to leading our facilities, he's invented two published computational sites, one of which he'll discuss today, the other a significant advancement on the use of 16S sequencing in the characterization of microbiome communities. Thanks very much. Rich, you want to take it from here?
Sure, thanks, Seth. Yeah, as Seth mentioned, at the end of the talk, if you've got questions and you just want to kind of capture them, feel free to type them into the chat, and Sarah will work to kind of capture those. We've got almost 75 folks on right now, so obviously it'd be a little tough to answer questions through the talk. But yeah, we'll definitely open up the mics as soon as the talk's done as well, so people can ask questions verbally if they would like. But if you're thinking of something and you're afraid you'll forget it, feel free to put it into the chat window and we'll try to capture those in the order they come in if you have any questions as we go. So today I'm going to be talking about the second generation of the CompBio platform that some may already be familiar with. We brought this tool to bear a number of years ago, and it has evolved through a period of the last 3 years. Quite considerably. So it's not just that we've added a few bells and whistles to it. There's actually a pretty substantial shift from the first version of the tool to the version I'm going to talk about today. Even the underlying engine itself is fundamentally quite a bit different and more powerful. So what I will do is I kind of broken the talk into 4 major categories. The first is a conceptual description of the PCMM and augmented artificial intelligence platform that the new system is built upon. So basically the foundation of the system. Improvements to the functionality and usability to the tool. So for those who've not used CompBio 1.0, I'll briefly kind of mention the basic utilization of the tool, and then for those who have used it, I'll be highlighting some of the key improvements that a number of labs have been asking for some time. In addition to that, then I will show the distinction from traditional pathway tools. I think when you see the context of traditional pathway tools and how they work and kind of how they stack up against a language processing tool like this that can effectively go read the literature, you know, with most everything that's been published within at least a month or two of the time you run your analysis, I think you'll be a bit surprised at how much more information that these types of tools can map. And then finally, I'm going to do talk about the comparative profile initiative. And I'm going to show you first how we are using this in translational research, how we're using it to compare everything from drug profiles to cell models to animal models to human disease. And basically kind of a request to the community to help us build up a central repository of publicly available data sets that everyone using the platform can use for this purpose. So just a little bit of background and evolution. I won't dwell on this for too long, but first, to those who have used CompBio version 1.0, this is a tool that we launched back in the 2018-2019 timeframe here at WashU. It's sort of what I refer to as the tricycle version of the tool. It was based on an engine called BKG, which was biological knowledge generation. It uses something we call contextual language processing to read the papers. For all the genes or metabolites that you would put into the system, and then it would build a sort of a holistic knowledge map around that. The technology behind the tool was licensed by Kingdom Capital and a company they spun out called Perchaya AI. But that doesn't affect folks here at WashU. The tool is freely available to all Washington University labs and investigators and always will be. But for those outside of Washington University, there is commercial access to the platform as well. By 2020, we had about 150 labs using version 1 here. It has gone into supporting work for quite a few papers, and I'll show a few of the key papers later on in the talk. But today, what I'm really going to talk about is version 2.0, which is a bit more advanced than the original version. That is based on this new platform. And I'm not going to talk about the third generation today, but I can tell you that the third generation is on the drawing board, and it's easily as far beyond what the current generation is as what it was beyond the first generation. So, but that's probably about a year or two out before we have that one ready to go. So for those that are not overly familiar with machine learning concepts, and for those who are, typical machine learning frameworks, especially task-directed networks, look a little something like this. You have some sort of data fabric where you have information, knowledge data coming from lots of different sources. You typically have some sort of layer in between that where you format the data, you scrub the data, you normalize the data, you have to have lots of observations, and then you have to label the data, at least, like I said, in these task-directed models. So if I want to basically have a model that can recognize a tree, I put in lots of trees, and that goes into my classification system. And so now when I come in with a new picture, I say, is this a tree? And if it was a good training model, it will, if it was a good training model, it will actually go through this and it will detect that it was a tree. I'm sorry, having a bit of a button issue there. And so you'll get the right answer, but there's a lot of limitations to this. So if you've got noisy data going in, you're going to get noisy models. Your ability to characterize things won't be terrific. The models are often very task specific. So when you build a model, you ask a specific question, you get a specific answer. If you want 10 different types of, if you've got 10 different types of questions you're going to ask, you probably have to build 10 different models. And these systems lack traceability. You really don't know why it is. It says something is what it is or isn't what it isn't. I mean, you get a score back and some type of values that give you some indication as the likelihood of its classification. But at the end of the day, you really cannot trace the information back to why it made the decision that it made. So we rethought the problem. In biology, you can take the same data set and you can have 10 different people look at that one data set and want to answer 10 different questions. Having worked in big pharma for a number of years, I know this all too well. People in discovery might look at it for a new target. People in toxicology are looking for side effects. People in development are looking for the population that they should be taking the target into. And again, I mean, you basically got a lot of people asking a lot of different questions, potentially the same data. So a system that is so task directed isn't ideal. And while there are advances on this and things going in better directions, that really is kind of a basic overview of artificial neural network task directed machine learning type applications. So we said, well, what if we could get rid of all the upfront and let's replace this with something that, as far as we know, doesn't really exist yet. It's more of a human-like contextual retrieval memory model. What if we could store memories with all of the context associated with them and be able to retrieve not only on the primaries of the memory, but on the context as well? This is something human beings are very, very good at. We're not as fast as computers, but we can remember that we were somewhere someday doing something and a jet plane flew overhead that had nothing to do with what we were doing, but it was part of the context of what happened that day, right? And we're pretty good at making those types of connections with things. So we sought to build a memory model that could do this, where effectively what it can do is it can take many different types of data and information and ingest those as memories. And then within those memories, it can normalize across them, across those many different types of memories, identify context-specific signal within the knowledge that it gathers from those normalized memories, and then create contextual knowledge patterns of information from it. Now, it sounds like a mouthful, but the idea looks something like this. We would then next be able to take those and compare those contextual knowledge patterns with machine learning algorithms that are designed to look at knowledge that has been built that way without necessarily being built to answer specific questions. So in other words, if I could take two images of a tree and turn it into contextual knowledge, it would look something like what you see below in terms of how different pieces and parts of the trees are connected to each other, basically their context. And one of the things that changes in the falls is that the leaf and the stems associated with the leaves are connected to the soil instead of to the branches, right? So there's a change in context of that information. And so what we sought to do then was to have a machine learning model that could actually identify the similarities and the differences in the patterns of the knowledge without necessarily even knowing what a tree is. It doesn't have to have 100 pictures or 1,000 pictures of labeled trees to say the difference between this knowledge pattern of a tree and this knowledge pattern of a tree is the same or different in this way. Okay, so how do we do this? Well, this is a super high level simplified view. Let's say you've got the information associated, and again, one thing I want to point out, we don't do image analysis. I'm simply using these images for context. But if you took the knowledge associated with a family having a picnic, right? You could have something in the old days of the triple store, it would be family has picnic. But what about all the context? There were trees, there was grass, they were sitting on a blanket, they had all kinds of fruit, there were flowers, there was a soccer ball sitting on the grass. There were all these different components of context associated with that event. And so what the system we built does is it takes all recognizable concepts from a knowledge source. Assume this is a knowledge source, this piece of information about a family having a picnic. We take all of the concepts that we understand and basically create dimensional categories for those concepts. And so then we can store a contextual memory about this picnic. We can store all of the context that went with that family has picnic assertion, if you will. But then we can take it even a step further. Once we've assembled a number of core memories around knowledge sources that contain information about similar sorts of things, we can take it a step further and we can start to build contextual knowledge, meaning that I can identify signal between different concepts that occurs in a non-random way. And this is what the system does. It breaks everything down into its basic components. It has dimensions for many, many aspects of biology and medicine, not plants, foods, and people and objects. I'll show you in a minute what our dimensions look like. But then once we have a number of core memories, the system in a very generalized way can identify signals between any of these concepts that have non-random interactions. So in a real world application, it looks something like this. What we've done is we've taken 30 million abstracts and 3 million full text articles that we have access to from PubMed, and we've ingested those into a core memory model. And so we have these millions of knowledge sources of information. But like I said, in a given abstract, what one person finds interesting may be completely irrelevant to another, and they may find something else interesting. So it's literally all of the information that we think may have biological relevance from an abstract or a paper that we store into these core memories. And then the core memory model becomes persistent, meaning it stays in the memory of the computer and it basically acts like a service. And so as you can see, we have many different dimensions that represent things that are interesting and important to biological sciences. Entities such as genes, metabolites, microbes, physiology, drugs, diagnoses, procedures, on and on. We have many, many different dimensions that are represented in the system. Some of them are linear and some of them are actually hierarchical, meaning that we can aggregate information across the nodes in those dimensions based on the hierarchy. For example, we use the drug hierarchy so that you can not only see information associated with Lipitor, you can see information potentially associated with the generic atorvastatin or potentially even all H&G-CoA reductase inhibitors. But now let's take the example of a gene. So a gene is a coordinate in in our entity dimension, and we will have many memories that may point to IL-6 along with all of their context that coming from all the papers that talk about IL-6 or some form of the IL-6 gene or protein. So we create what we call an aggregate memory for that coordinate.
Excuse me,
then,
Rich.
Yes.
You can use your cursor if you like, just to point onto the screen.
Oh, sorry, I was pointing at the wrong screen. So my apologies. So you can use your, basically you can aggregate everything from the IL-6 gene into an aggregate memory. But then in order to know what signal, right, what is meaningful about IL-6? I mean, I might find the term microarray 10,000 times with IL-6, but so what? Microarray shows up with every gene that's ever been put on a microarray. So who cares, right? So we use this, we compare it to its peers to identify the fingerprint that is IL-6. This basically allows us to identify knowledge that is signal containing. This is what we refer to as the signal detection layer. And it's generalized across all dimensions. In every single dimension, at every level of that dimension, even if it's hierarchical, we can create these normalized fingerprints for those coordinates. Because it builds the connections up based off of signal from the underlying sources, you can almost think of the PCMM as an inverted neural network. We've effectively turned the network inside out, and instead of having all the edges and removing ones we don't find signal in as we train, it's literally just building edges up as you add more and more knowledge to the system. And it's a sparse edge system, meaning it only keeps the edges that it believes contain signal. So in effect, what we put together with the PCMM and the machine learning is, like I said, conceptually kind of like an inverted neural network with a neural network. One for memory storage and retrieval, the other for processing of complex patterns. So as I mentioned, the Assertion Engine, what it does is it takes, as an example, again, a very simplistic example, I've taken a bunch of representations of knowledge about trees in different states or components of trees in different states. This is what our system, this is what the PCMM can build. What the Assertion Engine can do is it can take these and compare them to each other, as I had mentioned previously, and it can identify where contextual patterns have been preserved and where they've been broken. So in other words, it would identify that, yes, this component between these two tree images has been preserved. The leaf stem component has been broken because the context has changed. Leaf and stem are still in context of each other, but their context is different between these two pieces of information. And as you can imagine, this is a very simplistic example. This doesn't take, machine learning necessarily to be able to solve it. But when you take patterns, biological patterns with thousands of concepts, from thousands of entities that you started with and across many, many dimensions, identifying these preserved patterns requires something more along the lines of machine learning. Okay, so that's the conceptual overview of the system. So the old BKG engine that just used contextual language processing to basically read the papers on the fly as you put them in has been taken out, and this new engine has been put in. And I'll kind of explain a lot of the advantages of that in just a minute, but basically this is the way the system works. We ingest everything we can download from PubMed into the PCMM with its signal detection layer, and then the pattern detection layer, which is the Assertion Engine, sits on top of that for doing comparisons that'll actually show at the end where we compare very, very complex models. Okay, so this is what we refer to as an augmented artificial intelligence platform, this combination of traditional machine learning neural networks with this sort of inverted neural network memory creation and knowledge storage system. Now, within the system itself, it looks very much like the old CompBio version 1. You take your omics data, whatever it may be, genes, proteins, metabolites. you dump them into the system, you give it a project name, but now the system runs against this engine and uses that to create the holistic knowledge maps that are integrated, and you can interact with them. So the basic functionality of CompBio version 2 looks very much like CompBio version 1. I mean, the basic interface is very, we wanted folks that were used, so we've got 150 labs using it at WashU alone, not counting people that are using it outside of WashU. So we wanted to make sure that it wasn't too different in terms of the feel. But the tool allows you to see the data at many levels. So you get the kind of the holistic map back, which gives you themes, which are connected to themes that are close to them in context. So the terms and the biological processes they tend to represent are often very closely connected when they're positionally close to each other on the screen. You can drill into those to see how the concepts within those themes are interconnected. You can click on a theme to see all of the entities that are connected to the theme. And then you can even see the enrichment score for that particular entity with the different concepts in the theme. So for example, SLC 9A3 has like a 450-fold enrichment for microvillus as compared to anything that you would find by random chance. So this not only tells you what entities are associated with this biological process, it actually tells you how strongly connected and associated they are with that biological process, which is something you cannot get in pathway tools. They simply compare lists to lists. What's new in version 2.0, there are actually a number of upgrades. So one, we've really cleaned up the entity types. You can still put in the entities you could put in before, but we've put a lot of effort into really cleaning up a lot of the stuff that was out there that's questionable or very ambiguous, very hard to disambiguate, that sort of thing. So we've done a much better job of cleaning this up. Microbes are still limited. You still have to input a genus species as a naming convention. So we're still a bit limited on what we can do with microbes, but there is some ability there in the microbe space. But the rest of these are fairly comprehensive. We have really most of the mammalian proteins, gene protein space. We have about 35,000 metabolites at last check, which include all the known drugs and even a lot of things that have never even made it to clinical trials in some cases from drug companies. Within the engine itself, it's much faster. Runs before would take 30, 40 minutes sometimes. They now take 2 to 3 minutes on average. Again, because everything is contained within memory. The big advantage is we have vastly improved normalization because everything runs in memory. The normalizations prior in version 1 were approximations. We basically did lots of prior normalizations for different sizes of entity lists. And when you started to get into large numbers of things other than genes and proteins, the approximations got weaker. That's not the case anymore. When you put in a list of, say, 400 genes and 100 metabolites, at runtime, the system creates 5,000 random sets of 400 genes and 100 metabolites to compute the underlying statistics in order to determine what is significant and what is not. So you have complete normalization, much, much more accurate, vastly improved, and true multi-omic. It does full multi-omic based on what your input list is. And so you're coming, your data coming back, your signal is much, much better than it was before. You don't have to put in numbers of concepts anymore because the system actually figures out the optimum number of biological concepts that represent the biology of the data you've input. And we're doing a much better job of handling ambiguous gene names in a lot of cases. There's still a few out there that we hit every so often. We'll hit an ambiguous gene name that we didn't pre-validate correctly or something from a paper. So once people find those, we ask them to send them to us, but they're very much the exception now and not the rule. The knowledge map itself looks very much like it did before, except you'll notice a few differences right out of the gate. One, you'll notice that a lot of the themes that come out with their default, just say theme 1, 2, 3, 4, based on their theme scores, have stars. And that means they have suggested annotations associated with them, meaning that the system is able to look at the concepts and based on some of its underlying knowledge that it's ingested, determine what the likely annotations or possible annotations for that theme are. That makes annotating these themes much faster than it used to be. Usually 2/3 to 3/4 of the themes will come back with potential annotations that you can click from, and they're sorted by their score. So this really helps with the annotation process with the themes. Another thing you'll find is that, and this is very new, we now have theme level p-values and normalized enrichment scores that are very similar to what a lot of folks are used to with GSEA. So the normalized enrichment score calculation is done pretty much exactly like it's done in GSEA, where you take the score of all your random data sets and take your mean over that mean, and you get a normalized enrichment score. The p-value is an empirical p-value computed from randomized distributions of entity lists of similar size within a certain range of the one that you're looking at. And so you now actually get back p-values, and you're going to see how useful those p-values are when I show you some of the real world data that we've processed through it. And then finally, as I mentioned before, the first version only had abstracts. We now have full text articles in the analysis, and the tool scans through blocks of text within full text articles to generate the final sets of memories along with abstracts for the information. And you can drill into, again, the text articles as well as your abstracts to do your assessment of the biology if you so choose. There's another completely new tool in the platform. It's called BioExplorer. BioExplorer is basically a literature mining tool. So we realized one day, it's like with the PCMM now and all of these normalized aggregated memories pre-built, any concept, any gene, any physiology entry that we have, if you want to see a CompBio-like map with concepts and biological themes are built around it to show you what's out there in the literature, what's known in the literature around that idea, you can just put them in and it will generate a map very similar to CompBio. You can put in concepts, pathophysiology terms, physiology terms, all the entities. You can do a single term query. You can create multi-word queries, which is like an and, or you can do multi-term queries, which is like an or. And when you put those in, it will go off, and you can even tell it, do I want to pull these from concepts or entities? Do I want to include physiology terms? You can even tell it what you want it to include in its analysis, and it will build a CompBio map. And much like the CompBio map, you get starred themes that you can annotate. You can click into it. You can see the concepts. You can drill right down into the papers. For example, I just put in coronavirus and ACE2 for this one. And basically, you can see a bunch of stuff that comes back around SARS-CoV-2 and, a lot of knowledge that's really been generated within the last year. So this is a nice tool. If you've got an idea or concept you're not familiar with, it's a nice tool to be able to dig in and kind of get a nice landscape of what's known in the literature about it. Or if you find something in your data set, some idea, some biological process you didn't expect to find there, you can come over here and put that in and see if anybody else has ever talked about that process in the context of something similar to your experiment. So it's a good way to do a nice pull of information and sort of logically sort information from the literature. Okay, so lots of new functionality in the tool, lots of major enhancements to the accuracy of the tool. How does this compare to stuff that people are used to using? So we took two comparators. We could have taken more, but there are so many tools out there. It's like it just takes it takes a while to do these things. So we picked a couple that we know a lot of people use and are familiar with. So Ingenuity IPA, the Ingenuity Pathway Analysis tool, been out there for a long time. A lot of people know about it. Been lots of publications. Nice interface, long history, and it costs a lot of money. Enricher is a free tool. It is academic in nature, and I'm going to just put it out there. We really like Enricher. It's a really nice tool. It's got like 90 different types of knowledge bases under it. So, I mean, it's even got like transcription factor related knowledge bases and stuff, like goes way beyond pathways. So it's a really nice tool. And so we like to use it when we want to kind of do a broad sweep of knowledge bases. And so, kind of giving Enricher a little bit of a plug as a traditional pathway tool. But the other thing that's nice about it is because it has so many knowledge bases underneath of it, we could use it as a way to show how many knowledge bases you need to start to include in order to get results similar to what comes from CompBio, and at the same time, how much of a data inflation problem you run into when you do that. So we did a number of different comparisons, and I'm going to go through those now. So the first thing we did is we did kind of a positive control test. So we took 31 entries that were kind of randomly pulled from Go. Why 31? I don't remember. We just pulled a bunch, and because we knew we'd have to go back and we'd have to do a manual comparison in IPA and Enricher and some of these different tools with all these pathways to see if, if it's not an exact name match, at least it was close. So a human being had to kind of go in and say, all right, is there at least something close in IPA and Enricher that kind of matches these ideas? For CompBio, it's a little easier because it's concept-based. It's very easy to search on those concepts and say, is it there or not? So we did this with 31. We pulled Go out of the Enricher knowledge bases. It would have been a little unfair to take 31 Go pathways and then put Go in as one of the knowledge bases that we use for comparison. So we just used 7 different pathway databases. We use NCI Nature, Wiki, Panther, KEGG, Reactome, Biocarta, and SIGDB. So, some pretty commonly used pathway knowledge bases for Enricher. And then IPA, we use the traditional tool background for that. So what you'll see is that of the 31 Go pathways, there were 23 that had a good name match theme from CompBio, but 30 of 31 actually had the concepts or a strong concept match, even if the theme wasn't named or pre-named, matching the pathway or ontology name from Go. So pretty good hit. It pretty much gets just about everything back in one form or another. Enricher did pretty well also, even without Go in there. 25 of the 31 had something close or something in the ballpark of the named Go pathway or process. But keeping in mind that this came from 7 different pathway databases in order to get that information. Ingenuity did pretty poorly. And this really surprised us at first because Ingenuity has Go as part of its background. So why Ingenuity couldn't even get half of the Go pathways or something close to the Go pathways, where we took the genes from that Go pathway, dumped those genes in and said, just give us back anything adjusted p-value significant, and it didn't even get half of them. So that was certainly interesting. And what you're going to see is that is not an outlier. That turns out to be the case quite often for the comparisons with IPA. The next thing we did was, okay, fine, positive controls look pretty good. But you know what, if I take 7 or 8 different knowledge bases, maybe I can get something close to what CompBio gets. Granted, I got to, kind of work my way through all those outputs and results, but maybe I could get something close back. All right, well, what happens when we put in real world data sets? When we put in real world data sets, what you will see for these pathway tools is their results are all over the place. CompBio's results are actually very, very consistent and surprisingly consistent. So first thing I'm going to give you is a couple definitions. So first of all, within CompBio, the number of themes that were selected were based off of 2 things. They were based off of p-value significance that I showed earlier, and also based off of the information content plot. So, and we're going to be adding this plot to the tool itself within the next week or 2 before the formal rollout. And what this shows you is the number of genes that are mapped uniquely by theme. So in other words, it says in this case, 60% of the genes in the input data set had mapped by theme 2. And by the time I got to about theme 10, almost 80% of the genes had been mapped to at least one theme. Okay, so what you can see is that over time, you just basically start getting, as you add more and more themes, you're basically just adding more and more mappings of biological information onto the same genes. You're not getting new gene information content into your biological processes. And therefore, once you start getting past that, and we know this from utilization, you start to run into a lot of redundant secondary, tertiary type of biology events associated with these genes. They're not wrong. They're just not central to the experiment that you did most likely. For the other 2 tools, we use their standard adjusted p-value cutoffs. in order to get the information. And as I mentioned, in this case, we allowed Enricher to use all 10 of the knowledge bases that we started with, including the 3 most popular Go ontologies, including biological processes, which a lot of people use. It's fairly large, incredibly generic, includes a lot of stuff. So 3 different data sets, and these were all published data sets. One is the Crohn's disease data set from the Stapp and Beck lab from 2018. One is a CTCF conditional knockout mouse from the Milbrandt lab. These were upregulated genes. This cluster 5 was downregulated genes. And then a severe acute malnutrition study from Jeff Gordon's lab. And this is not, I apologize, that is an error. That should say proteins. So these were SomaScan data, not RNA-seq data for the Gordon study. So you've got 2 RNA-seq studies and 1 proteomic study using SomaScan. And what you'll see is that within the first data set, CompBio was able, within those first 35 themes, CompBio was able to map 74% of the input genes to at least 1 theme. And the data reduction parameter of basically taking 373 genes down to 35 themes was a 0.1. When you look at IPA, it only was able to map, again, 30% of the genes and its significant pathways. And there were 81 significant p-value adjusted pathways, but it only managed to map 30% of the input content. So very low information content in IPA. Enricher was able to map 67%, but that went across 243 pathways. So basically I input 500 genes, 338 of them, 67% mapped into 243 pathways. That's almost not a data reduction. You've got almost as many pathways to sort through as you have genes that you started with. So if you're trying to understand the data, if all you're trying to do is make the obligatory heat map for your article to go out to whatever journal, okay, fine, it probably doesn't matter. But if you're actually trying to understand your data, this isn't much of a help when you don't reduce the number of genes to the numbers of pathways very much and processes. Now, granted, there's a lot of redundancy in here, but obviously that makes it even more confusing, right? You have to go in and remove all these redundant pathways with somewhat similar names to kind of figure out what the unique content might really be. On CTCF, the results were even more dramatic. CompBio, again, maps 75% of the genes within the top 35 biological themes. IPA, only 27%. And even Enricher, with 10 different knowledge bases, was only able to map 33% of these. Now, whether this was because it's mouse and not human, I don't know, but we were really surprised that even Enricher had a relatively poor information content score in terms of the numbers of genes mapped to processes. So, you can see in these 2 cases, CompBio has actually really outdone the other 2 by quite a bit. The SAM children was a little different in that you did actually get better. You managed to get a 74% map back with IPA, which was good. And these are coming from proteins that are on the SomaScan platform. So they're probably mostly very well-known proteins, which is probably why you have such a good map back rate for these. But even then, you still got 74 as compared to the 90s for CompBio and Enricher. The big dramatic difference is for Enricher to get to 96% map, it had over 2,000 pathways that those genes mapped into, which is a huge data inflation, right? So, I mean, the amount of work you'd have to do to kind of figure out what was meaningful and relevant biologically is quite large. So you can see in these real world data sets, sort of the maximization of gene information content to minimized amount of biology explaining those genes. The tool just dramatically outperforms pathway tools. And again, it's because we're looking at everything in context, and pathway tools just take a list and compare it to another list. That's essentially what they do. And you can just kind of see the overall numbers down here, and it kind of bears out in which I showed in 3 examples. Now, the next question may be, well, how right is any of it, right? I mean, that's great if it's mapping all this stuff, but what if it's all wrong? What if it's all garbage? So what I'm showing here is an example of a concept entity assessment with validation and traceability. This is another thing where there's a number of things you have here that you just don't have in a lot of those other tools. So we took a particular theme, theme 7, which is very clearly, if you look at the concepts, talking about adherence junctions, and that was the top auto annotation. We then took 3 of the concepts from this: adherence junctions, adherence focal adhesion, which are all super strongly associated with the main theme. There were 64 genes enriched for the key concepts in here. Okay, so, and in addition to the fact that there are 64, you can see the enrichment score for each gene to that particular concept. So, I mean, these are huge. Even in the 30s, these are like 30, 40, 50-fold like enrichments, 100-fold enrichments, 1,000-fold enrichments. These are super enriched genes around these concepts. That is not something you can get in a pathway tool generally. Generally, when you look at pathway tools, it says, here's the list of genes associated with this pathway. You have no idea what the relevance of those genes are to the pathway. You know, some human being put it in the list. And unless you're going to go look at those papers to see what that relevance was, you have no clue. Within this tool, within 2 clicks, you can actually see what the papers are that we're pulling in to make the associations. So again, comparing the 3 tools, CompBio has 64 genes associated with these. We looked at any pathway from enricher or IPA that had anything to do with adherence junctions, even anything close. I think with Ratasian pulled like intracellular junctions or something like that for enricher. And you can see enricher got only 28 genes mapped and IPA 9. And if you kind of look at the Venn diagram, what you can see is CompBio gets the vast majority of the genes for the other 2 tools. We only miss 1 of IPAs, and it's kind of questionable as to how it's linked into the tight adherence junctions. And we get 21 of 28 from enricher from the 10 different knowledge bases, and then 33 unique. And so if you look at some of these, you say, okay, yeah, but again, what if those aren't right? Well, again, you can go through and you can see a lot of these are junction proteins just by the names. But MyZap, we just took one right off the top, for example, is an adherence protein. It's what it is. It's what it does. And yet the other 2 tools have no information mapping that into adherence junctions. And if you go drill down by clicking over here on the gene with the theme, it will show you the papers linking that gene to that concept. And you can see for yourself how tightly connected it is, and the fact that MyoZap is in fact an adherence junction protein. And a lot of these proteins are, if you dig in, you'll see are actually quite well linked to adherence junctions, often in epithelial cells. Okay. Another question that we always get with the tool is, well, how many genes should I put in? Should I use nominal p-value? Should I use the really stringent adjusted p-value stuff? Because a lot of tools aren't very good at detecting signal within noise. And I'm afraid if I give it a lot of noise, I'm going to get a lot of noise back. And if you're working with pathway tools where you're comparing one list to literally tens to hundreds of thousands of other lists, yeah, that can happen. But CompBio, as you've seen, works in a completely different way. It takes that list you've given it, it basically reads all the literature around it and starts building the signal up contextually from what you've given it. And then based on that and our comparisons with random data sets and so forth, it is able to very effectively tell what signal. So what we did as an example of this is we took a data set we had from the adult Crohn's set. The cluster 5 had about 1,000 genes in it, and it was just by chance, it was this very, very nice breakdown. The 1,000 quote unquote significant genes, nominal p-value, not adjusted p-value. The top 500 had a p-value less than 0.01. The bottom 500 had a p-value between 0.01 and about 0.05. So it was just this very nice fortuitous breakdown of the genes. So we took those 2, and so no, there is no overlap in these 4 sets that we analyzed. The genes are unique in each of the 4, top 500, bottom 500 by nominal p-value. The next set we took were the first 500 that were not significant, greater than 0.05, but less than 0.1. And then finally, we took a purely random list of 500 genes that were just literally generated random, had nothing to do with this data set. And what you can see in looking at the theme p-value trace down below is that the top 50 themes for the top 500 are basically all, they're less than 0.001. So they come out basically just right on top of 0. The bottom 500, completely different list of genes, but between 0.01 and 0.05, also almost completely almost near zero significant. There are only 2 that are slightly off, and those are even under 0.05 in terms of significance. What's interesting is when you look at the non-significant list, you can see that the first 10 to 12 themes, there are about 10 themes in the first dozen or so that are actually quite significant. And when you go over and look at the normalized enrichment scores, you actually kind of see this pattern in terms of the overall enrichment going down. The gold bar represents random, and you can see the p-values are, you got one that maybe gets close to 0.1, but other than that, they're terrible. And you can see that, as you would expect, when you look at a random data set, all of the themes are basically just hovering around 1. Again, the first one slightly above, but not by much. So you can see, even in this non-significant 500 list, the tool thinks it's finding signal. Now, the question is, does that signal have anything to do with the true signal we know is in the top genes? And the answer is yes, it actually is. So we use the Assertion Engine, and I'll show you in a few minutes what these numbers actually mean. The Assertion Engine, the global similarity score is a value that goes between 0 and 1. And really anything over about 0.06, 0.07 is where they start to become significant, meaning that the tool believes there's significant overlapping contextual biology between the data sets. And you can see that the bottom 500 score is quite significant with a good overall similarity score of about 0.13. Even the non-significant is close to 0.1 with a very significant p-value, indicating that there is overlap in this signal with what you see in the top 500 gene list. And in fact, if you go into the map, that's exactly what you see. These 10 themes are basically almost line up with the top 10 to 15 themes in the top 500 list. The tool has gone into a list of genes that's very heavy in noise, but because of the contextual analysis was able to determine there was actually some signal in there, and that signal matches with the most significant gene list. Okay, so that's a bit on the comparison of the tool. We have a number of labs, as I said, who've been using this. There are some nice papers and some very high impact journals that have come out using it. In fact, fingers crossed, there's a Nature paper that's, I think, very close to being accepted in the Gordon lab that actually is profiles or shows a very nice heavy use of CompBio in their analysis and their determinations. But again, we've worked at different types of data. Some of these data are proteomic, some are transcriptomic. We've worked with GWAS data. Again, we're working with metabolomic data. So just about any omics data can go into the tool. Okay, now in the last bit, I'm going to tell you about the Assertion Engine. So this is where most of the machine learning comes in. And this is where we have the ability to start really doing almost high throughput translational biology. So the Assertion Engine interface is fairly simple. It kind of looks and feels like the user interfaces for CompBio. But basically what you do is it will show you all CompBio projects that you've built that are in your directory. And it will say, tell me which projects you would like to compare. So you give it a name for your project and you say, okay, here's the project I want to compare to, and here's the project I want to compare. And you say go and say generate an assertion. The other thing you can do though, is if you have a whole group of projects, and the example I show here is exactly that. I have 3 different animal models that are somewhat related to Crohn's disease. So I've taken all 3 of those and I've created a library. And what that library means is for the project, I just entered the library name. And what the Assertion Engine will do is, in this case, it compared the adult Crohn's data set to all 3 of the animal models. And basically, it's going to give me a profile of what those animal models did similarly to that preserved human profile across the 3. And in order to see that, when I go in and I look at the result of the Assertion Engine, you can see here, the ACD, full text, this is the adult top 500 genes I was just talking about. This is animal model 1's downregulated, and there's its score, and there's the p-value. There's animal model 2's score and its p-value, and animal model 3's score and its p-value. And if you really want to get a good feel for the p-value and in an empirical sense, in terms of how far away from noise the data for your data set actually are, you can click on that value and it will show you the full distribution of the randomized runs against non-random data sets. It will show you the comparators that it uses, and then it will show you where your data set sits. And you can see in this particular case, this data set sits well above even the highest random entry that was run for comparator purposes. Then, if you want to see the actual biology that overlaps and how it overlaps, you can click on the value itself. There's a dendrogram down here too that kind of shows you how the things group. But if you want to see the comparison itself, you click on the value and it will actually show you this, what looks like, if anybody's ever played the old game Risk, kind of looks like a Risk country map. And what it is a flattened 2D projection of concepts. You can kind of think of it as, it's not quite the same thing, but it's almost like you take the 3D projections in CompBio and flatten them down into 2D and then show the concepts within the countries. Now, you won't be able to read this on your screen, but one of the things that you'll see right away is, again, these countries are mapped out projecting everything that this had in common with any of the three comparators. And so you can see that mouse model 1 tends to have most biology preservation on the biological processes to the left of the map. The middle one actually is much more conserved to the right half of the map, and the third one is more conserved through the middle of the map, indicating that while there is conservation of biology in the three animal models to the human disease, what is preserved is different. And you can actually drill in and see these, and I'm going to show this to you in a second in a real world example, but I just want to kind of show you the interface and how you can use it. So as a real world example, we have, and I got to give credit to Allison Mayer from Perchaya, who actually assembled these data sets. There are 8 human NASH and NAFLD, so NASH, non-alcoholic steatohepatitis, and non-alcoholic fatty liver disease. And there are 8 human data sets that she collected in these categories and 2 mouse data sets. And these were all publicly downloadable RNA-seq data sets out of Geo. And she created upregulated and downregulated gene sets because she kind of wanted to do apples to apples comparisons. And we took those gene sets and we created CompBio projects for all of them. And then we created a library out of all of them and we compared the library to itself. And so within that, we got the cross-compared global scores and local scores for similarity in all of them. And what you see below is the top half of the browser window that you get back from that result. And you can see, obviously, anything ever compared to itself gets a value of 1. No surprise there, as it should. Don't even bother putting a p-value there. And you can see these are 3 human data sets. 2 of them are NASH versus healthy, and 1 of them is NASH class 1 versus class 3. And when you look at the human data sets, you can see their scores. There's quite a bit of significant overlap in biology, and the scores are pretty good overall in terms of comparing all 3 of those, even though the one is a little bit different in terms of what it represents. But then you go down and look at the mouse models, and you can see there's even some significant, not as good, but there's some overlap between the mouse models and the human. But then when you look at the mouse models compared to each other, you can see that they're almost kind of off the charts in terms of their biological similarity, even though one is NASH and one is NAFLD. This was kind of interesting. So we were intrigued to see what that biology represented. So first, though, we wanted to look at the human data, and we wanted to say, okay, we've got 3 different human data sets. We realize one's maybe slightly different than the other 2. Let's kind of look at how the human data sets are similar or different based on their biological overall biological context. So I picked the Duplessis data set as the center one just because its scores were most central to the other 2. I could have picked any of the 2 because you can click on these as you go across the screen and see these maps, but I took this one just because it was the most central. So this is the map for the Duplessis data set. This is all the biology that maps very nicely across the other data sets, okay? And meaning that it's not necessarily everything in the Duplessis data set, but it's everything in the Duplessis data set that shows up in at least one of the other data sets. And because of that, as I go across the row, it keeps the country formations, the 2D projection, the same, so I can do a direct comparison. So the first thing I want to do, because there's a lot on this screen, and I don't want people to have to kind of look through all of this, I'm pointing out some of the key stuff. One of the things that you'll notice right in the middle, CompBio doesn't know this is NASH. CompBio knows you gave it some gene lists that you wanted it to run, and they were anywhere from 80 to 200 and something genes. And yet, non-alcoholic steatohepatitis shows up as one of the most central concepts in the entire maps. Basically, it seems to think that everything is centered around that. And right next to that, you can see fibrosis and matrix remodeling. On the other side, basically fatty liver. Very good to see these things and very tightly connected into the NASH. And then you can see inflammation and sugar metabolism take up heavy components of the maps to the sides of that. Not unexpected. This is great. This is what you would hope to see when looking at data sets like this. But how do the other data sets compare? Well, when you look at the Aaron's data set, you'll see that the NASH, the fibrosis ECM, and to some degree, the adipose fatty liver, lipids, fatty acids are pretty well preserved. And you see that by the fact that these that the names, the titles are all strongly dark, and you see a lot of connecting lines. That means the contexts are preserved between these data sets. It's not just looking at what's in the list, it's looking for their contextual preservation. What you may notice, though, is there's not much inflammation in the Aaron's data set. Not a lot around sugar metabolism either. And so very quickly, we were able to see some distinct similarities and differences between these two. And if I look at the Freddy's data set, similarly, what I find here is while I see more inflammation and sugar metabolism, again, NASH still very central, the fibrosis is almost gone. And the ECM is almost gone. So even in these three human data sets, while there is this clear centrality around NASH and some of the key themes and concepts, there are clear differences as well. And this is a way you can see those differences and similarities very quickly. So what about the animal models? Because you saw those huge scores, these scores approaching 0.28, p-value is really kind of off the chart on these. So here's the Zhang model. This is the actual NASH model. The other one's NAFLD. And again, I'll just highlight these things so you can see them fairly quickly. Right away, you can see there's something very different about the animal models. NASH is in here, but it isn't central. What is central is inflammation. There is a huge amount of inflammation in these animal models. And the NASH is really kind of a very small concept off to the side with a little bit of fibrosis and a little bit of fatty liver associated with it. And what's also interesting is the ECM, while there's a big component of it, it's not connected to fibrosis. It's in the middle of the inflammation. So the context of the ECM is different. It's in both, but it turns out that it's contextually different. And you can see that when you compare it to the other mouse model. ECM still there, still heavy, still a huge amount of inflammation. This one has almost hardly any. It still has the steatohepatitis and fibrosis, but very, very little. And it's just heavily, heavily inflammatory with a lot of matrix remodeling due to the inflammation. And so you can kind of see now how this can tell you and inform you translationally how a model might work or not work around a target and how relevant that path might be in a given animal model as compared to human disease data sets. And just for comparison, you can see here, this is the human data set now compared to this animal model. And now you can see there is stronger representation back over here with NASH and fibrosis, a little bit with adipose. But now you can also see the difference in that a lot of the lines connecting with ECM are going back to fibrosis. So really kind of giving you a hint is the contextual difference of the matrix remodeling in the animal model versus human disease. So with that in mind, we're putting kind of a call out to all of our colleagues that either use the tool or are interested in helping to generate this. We would like to create a repository of comparative profiles around drugs, cells, animal models, and human disease. And particularly, we want to do this in areas where researchers are doing a lot of R&D at the WashU School of Medicine. So, obviously neuroscience, inflammation, cancer, et cetera, metabolic diseases and so forth. And so we're looking for help from domain experts out there to help us identify these data sets. If you know of data sets that are sort of seminal data sets for human disease or animal models that people use a lot in your area of research, particularly mammalian animal models, we would like to, if you know where the papers are at, or the geo number where these data sets can be downloaded, we'd love to be able to start downloading these and ingesting these in and creating these libraries. As you can imagine, we'd have, a NASH, NAFLD library. We would have, potentially a rheumatoid arthritis library. We would have an AML library and so forth. And again, we would have animal models that could be in those libraries or separated so that you could compare animal models overall or within that category and kind of do this on a large scale. And then the idea is we would make these central because these are publicly available data sets. So when you bring in your private data into your account, you can compare your private data to these central repositories and the results end up in your account. So you can basically compare any data set you want, even the ones that are your private research, to these publicly available data sets. So we're looking for all the help we can get to help us assemble these sets. Again, we'll take papers or geo numbers, but if labs have already downloaded some of these data sets and used these, and you've already isolated the genes or proteins or metabolites, that's even better because all we got to do is take those and load them into the tool and make the library. So we would really appreciate any help anyone can give us in getting the word out that we want to try to build these central repositories for all WashU investigators to be able to access and use in their research.
Excuse me, Rich.
Yes.
We have a question concerning that came in. Do you have a kind of a checklist or guidelines that allows for high quality projects to create high quality database from that?
Well, that's what we're asking for folks who are domain experts to pass us or send us the high quality, what they believe are the high quality data sets in those areas. The ones that people tend to use, the ones that get referenced a lot, the ones that the animal models that laboratories tend to use. That's why we're going to the domain experts and asking for their help with this. We don't want to put in a lot of garbage data, obviously. And so we'd really like to be able, that said, define garbage data. If you have a mouse model that came from an impact factor 4 journal and it has a 0.3 overlap with human disease, was that garbage? Probably not. So the tool can also kind of tell us what's garbage and what's not. If you've got something that sits off by itself, doesn't seem to map to anything, doesn't seem to have any relevance to human disease, in effect, the tool is going to tell you what's good signal and what's not, right? Okay, in terms of rollout, so we are accepting account requests for version 2 right now. Anybody can request an account at any time. Just so you know, though, the official launch date for version 2 is probably going to be May 19th, which is 2 weeks from today. We just have some small updates and some minor bug fixes that don't really keep you from using the tool, but we may have to take the tool down from time to time in order to get those small fixes up. So just be aware of that. If you get an account now and you come in, 2 days from now and the system happens to be down, it's probably because we're doing an update. Users of CompBio version 1, as I said, we've got about 150 labs that have been using version 1. If you want to migrate from version 1 to version 2, your projects, we can do that in mass. If you want to do it yourself manually and just have it recreate them, that's fine. But if you want to keep the old projects so you can see how that compares when you rerun it in the new platform, we can do that as well. And we can kind of move those accounts in mass if people would like us to. We just need to know if there are labs that want us to do that. And then finally, training. So there are training sessions that we're working on coming up with scheduling now. We will probably have the first training session for this around the end of May or early June, about 2 weeks past the official launch. We're going to start having 2 different sessions. We're going to have sort of basic training for use of the platform, and then we're going to have advanced topics. So basically, I want to create libraries of animal models, and I want to compare human disease to my animal models, and how am I going to do this, or I want to do this kind of advanced, so basically advanced research topics in utilizing the platform. So we're going to kind of break it into 2. We can do individual lab training as well. If there are labs that would like us to work with their lab specifically, those have to be scheduled, but we can usually do that for a number of labs. We're also working on some comprehensive online training materials. We have some, but we're going to start breaking them down into components so you don't have to go through the whole training manual to get to the particular thing that you want. And finally, just the acknowledgements. I'm 5 minutes over, but not too bad. A lot of folks have worked on this. Really want to shout out to Rotation and Chad, who've been my partners in crime on this from the beginning, even really kind of going back to early days of thinking about how to try to do things like this back when we were at Pfizer 15 years ago. a number of other folks in GTAC who've helped with the development. Also a shout out to our early collaborators. Jeff Gordon's lab has been great. They use the tool quite heavily. A number of papers they have coming out are showing the tool quite prominently. Ta Chang, been working with him, and Kelly Van Dusen, Thad when he was still here, a lot of work in the inflammatory bowel disease area, a lot of early testing, real world application. Doug Mann, we've worked on a couple papers there in the proteomic world, and Jeff Milbrandt as well, where we've done a number of animal models. So, and then the folks from CompBio who've done most of the work on the Assertion Engine, and then really the whole commercial team from Perchaya and Kingdom and Worldwide. So I will stop there, and I know we're a little over, but if there are some questions and people want to hang around for a few minutes, I'm happy to answer what I can.
Sarah, if you could unmute everyone's microphone, you're welcome to use your microphones, or if you'd like to type into the chat box, you're welcome to do that. Chad, Ratasia, if there was any questions that came in you thought maybe Rich would like to be able to additionally comment on, feel free to present them now.
Wow, okay, so there's a lot of questions in the chat. Looks like Chad and Ratasia might have been answering some of those as we've gone along. Were there any questions that anybody asked in chat that didn't get answered? If so, feel free to unmute, throw them out there, or if anybody's got just wants to ask their question verbally, feel free.
Yeah, Rich, there was one question where one of the people had asked if CompBio could identify missing information in the literature.
Identify missing information in the literature.
Missing links.
That there ought to be links, and yet there aren't.
If you had data, you might be able to do that. I mean, BioExplorer is just a literature mining tool. It just reports what's in the literature. If you had data that was finding, so this is actually something that we're looking at. I don't know if this is the question or not, but one of the things that we've been looking at is you can compare in the Assertion Engine, you can actually compare a BioExplorer map to a CompBio map. So if you have data and you find some biological processes or themes associated, say, with a disease, and you're interested to see if other people have made that same discovery, you can generate a BioExplorer map around the disease and compare the two. And if it looks like the information is unique to your data, your CompBio map, that may suggest that that has not been discovered yet, or at least that biology has not been written about in the literature yet. So again, I don't know if that was answering the specific question, but that is one thing that we've actually started to look at. And we have seen cases of that where we found some really cool stuff and some data, and it's like, wow, I wonder if anybody else knows about this. And we went and built a BioExplorer map, and nobody had seemed to report anything around the biology we had specifically found associated with that disease. Was that the question, or did the question have a different context?
No, that seemed like it was about it. It was specifically asking about knowledge gaps.
Okay. Yeah, if you've got knowledge to suggest something new, yes. I mean, CompBio can't tell you or BioExplorer can't tell you what's not in the literature, because BioExplorer is built on the literature, right? But like I say, if you have a data set you've run through and it's identified biology and you want to see if that's missing in the literature, yes, you can compare a CompBio map to a BioExplorer map to do that.
Rich, I had two questions. One was whether we can add other model organisms. And the second one was if you can elaborate on the positive control tests with the Go gene lists.
Okay. So the first one, the last one's probably easiest. What we did is we took 31 Go entries. We took the gene list from those Go entries, and we ran those genes as independent sets through the three tools. And so what you saw was that either at the theme name level or at least at the concept level, CompBio re-identified the original named pathway in one form or another in CompBio in 30 out of 31 cases. In richer did it also at a pretty good clip, like 25 out of 31. IPA, not so great. Only 15 of the themes were identified as an output for the input gene list that were given from those specific Go processes and pathways. So that's what that was, is it's like, okay, take something that's a list of genes that's known to be associated with a pathway in some tool somewhere and see if any of the 3 tools can identify it as being associated with that biological process or pathway without using Go, right? If you put Go theme in and you're using Go in the background, well, of course you should get it because it's looking at its list. But interestingly enough, IPA still missed over half of them. The other model organisms, that's something we can discuss. I mean, you do have some model organism ability in the tool. The gene lists that we currently use, gene protein lists we use, are currently centered on mammals. But as you know, there are a lot of those names go back to fly and worm and other model organisms. So there is some capture of other model organisms, but it is mammalian centric at the moment. Could we build something more centric to model organisms? Yes, we could. And it would really just be a matter of sitting down with investigators, kind of say, okay, what are the most interesting model organisms? And we could basically create a dimension with model organism genes and basically set that up as an additional entity component. So it's doable. Obviously, as you know, it's a bit of work because you got to count for all of the redundant use of gene names across organisms that are not necessarily talking about the same gene, but it is possible if we had enough interest.
There was one more question, which I think you can explain. Whether we treat all data equally, assuming that all published data are correct and reproducible, and can we identify poorly collected data with either the scores or the p-values?
So what we look for are contextually reproducible associations. So if you have 3 different laboratories that made some association between 2 ideas that were completely independent of each other and an impact factor 3, 4, 5 journals, that would be enough to get a minimal hit for that thing. And it's because we put emphasis on reproducibility, meaning that I see this context signal showing up in more than one place. And when you go in and you look at the papers that are associated with these CompBio concept entity associations, you will see that. You will see that these things often come from very disparate areas of research. And in my years of experience, I put more emphasis, and right or wrong, I put more emphasis on the reproducibility of event than what journal a paper was in. Yes, I mean, obviously, if it's all one lab and they're all in like really low end journals, yeah, you might find something that isn't been reproduced in another lab. But it's most of the time you will find that the things that we identify have been identified by multiple labs in different contexts. And so that's where we really put the emphasis on, is on that reproducibility and kind of less on the journals. Now, that said, we do have the ability to filter on journals. It's not something we've done yet, but we can do that. And if there is enough call for it as a feature, we could do that. We could actually have it to where you could say, okay, go build my map, but only do it on journals with impact factor 5 and above. And so if we had enough call for that, we could add that functionality in. But I think most people, as they go through and they see how a lot of these themes tend to come out of kind of disparate laboratories and biological processes, that makes them much more believable.
Rich, I have a question here. What does it mean when you say to normalize memories?
To normalize memories? Oh, so I can go back here real quick. Normalizing a memory means I am going to look at the things associated with your aggregate, but I'm going to compare it to a whole bunch of its peers. So in other words, what I really want is I want the fingerprint for IL-6 that makes IL-6 IL-6. I don't want terms that show up with every single other gene and protein in the proteome or genome, right? I mean, those things generally aren't very interesting. There's not signal in IL-6 and microarray, right? Because every gene that's ever been on a microarray is probably going to have the term microarray show up with it. So we normalize it by comparing it to its peers and saying, okay, I'm going to take these vector weights associated with the aggregate. I'm going to compare you to a very large percentage of your peers. I am going to identify the signal vectors that identify you, your fingerprint IL-6. And that is what we call a normalized memory. We've normalized the vectors, the weighted vectors for IL-6 against its peers. And those, the ones that come out above, well above 1, are the things that make IL-6 IL-6, and not TNF alpha or PSG3. So hopefully that answered the question. Okay, so we've had quite a few people actually stick around for the questions. If there's any other questions anybody would like to ask that we haven't covered, please feel to throw them out there.
So I have 2 questions. Mm-hmm. So first one is about the data format. So, so for this augmented artificial intelligence platform, mm-hmm. So we can input different types of sequences, but you didn't tell us what type of data format can we use it? Just the sequence raw data?
So what you will put in is names. Okay, so for example, I'm showing this now. So the input list is for entity lists for CompBio. So gene symbol, or we've got a 35,000 metabolite symbol list that we pull from microRNAs. As I mentioned, microbes is a bit limited. You have to put in genus, species for the microbe. But effectively, you've done some analysis, you've generated some data set, you've done rna-seq or proteomics or metabolomics or a GWAS study, and you've got a list of genes or proteins or some type of entity, and you want to understand what the biology is that's represented by those lists, whether they were differential, whether they were significant, whether they were correlated. That's what you put into the CompBio entries. So if you've got 150 genes, if you've got 1,000 genes, if you've got 750 genes and 250 metabolites that came from a plasma analysis in humans, that's what you put in, is those symbols. And for genes, we use the official gene symbol from Montre. For the metabolites, we have what we call an official symbol for those, and that information is available, and we can kind of show you the lists for those. So in other words, we tend to use, like for drugs, the generic name, so tofacitinib, but we would recognize Xeljanz in a paper. So if you put in tofacitinib, it will go find all the places that tofacitinib or CP690550 or Xeljanz is mentioned. But in your input list, you tell us it's tofacitinib that you're interested in, and then it will go out and it will find all of the ways that tofacitinib is named in the analysis. So you're basically just putting in lists for the most part that have come out of some type of primary analysis that you've done.
Okay. So another question. So based on your introduction, so I was wondering, for this platform, it is possible to discover some new genes or proteins. No. So this platform just based on the memory, maybe from some data from the database or something.
So in order to have a memory, some name of a gene has to have been mentioned in at least 1 or 2 papers somewhere out there for it to be a candidate for the system. So if you have a gene that's never been mentioned in a paper anywhere, based on what the tool currently uses as its core memory model, it won't find anything around that gene. So that's why, like in some cases, you'll see we'll put in like 500 genes, 388 of them will come back as what we call valid, meaning there were core memories for 388 of those genes. So in terms of gene discovery, no, you're not going to discover a new gene. That said, you might discover a new function for a gene that nobody knew about, because you may find that this gene is associated with biological processes and properties from some information, even sometimes indirectly, in terms of how it was connected in with other concepts from other genes in the list. And so you can discover new functions for genes within the tool with that type of an analysis. But the one thing I will say is we are not genes, and the one thing I didn't talk about is we are actually we're creating a PCM based off of claims data from payer systems. So Prachaya is actually using this very same PCMM to do fraud, waste, and abuse analysis from claims and EHR data. So we do have the ability to put other information into the system. I think one of the things that we don't have in there yet that we do need to put in there are variant names. So gene variants, SNPs and SNVs and other types of gene variants. And I know Rataja's been working on that. And that is something that we will have in there fairly soon as well. So that will be a new entity type that you'll be able to search against our specific variants as opposed to full gene names. But yeah, if nobody's ever said this gene exists or anything about the gene, then we don't have any knowledge about that gene to be able to report back.
Okay, thank you.
Rich, is there user documentation, instructions for newbies that exist anywhere?
Yeah, I mentioned that in the last slide or second to last slide. There are some documents out there, but they're based on version 1. We're working on updating it for version 2 now. They should be available not too long after the launch. They're basically online PowerPoint slides that you can click. And there's like help sections you can click and it'll bring up like the PowerPoint slides associated with the help. But then the training sessions we really recommend people, if they can, attend the training sessions or ask for training for their labs. Because online documentation only goes so far. And we find that, sort of human interaction is a better way overall to bring people up to speed on how to use the platform. Okay. Well, thanks to all the folks who stuck around. And we're almost 23 minutes past the end of the talk and still about 30, 40 folks on. So thanks again for the great attendance. And if you have questions that come up, please do not hesitate to email either myself or Chad or Rataja or Seth. We'll get the email to someone who will know the answer. And also, I think there was some information, somebody had asked about the link for requesting accounts and so forth, and we can get that out. So feel free to send us an email with any questions you might have about the tool or getting access or whatever.